Hello student, good morning. As we see in the previous lecture, the limit test and the impurities. In the this lecture, we are going the limit test for impurities as the stepwise. Impurity is defined as the presence of one substance in other substance in low concentration. This may be a organic, inorganic, microbial, dust or moisture. These impurities impart the pharmaceutical substance through, as we described earlier, like raw material, intermediate, solvent, improper storage, cross contamination, moisture content, microbial content, the chemical instability, and the storage container. So, impurity in the pharmaceutical, this presence may cause cumulative toxic effect, decrease therapeutic effect, change in the chemical or physical property, difficult in formulation, incompatibilities. It will change the odor, color, taste, and appearance of the substance. In that, the water is a rich source for chloride, sulfate, carbonate. Reactor material used for manufacturing are the rich source of steel, copper, iron, zinc, and lead. The limit tests are quantitative or semi-quantitative tests designed to control the small quantity of inorganic impurities which are present in pharmaceutical substance. The limit test for chloride, sulfate, lead, iron, heavy metals, and arsenic. These all are official tests designed and mentioned in individual monograph of the pharmaceutical substance in Indian Pharmacopoeia. The observation for the limit test impurity, here we use to check the turbidity and opalescence. In the limit test, the standard opalescence or turbidity or color or stain obtained by reaction of non quantity of impurity with reagent compared with the test opalescence or turbidity. The reaction of specified quantity of the test sample with reagent, limit test, comparative test in which both test and standard must be prepared simultaneously at the same condition. Limit test apparatus. The type of apparatus which are used to perform the limit test for impurities. In the limit test of chloride, sulfate, heavy metal, and iron, Nessler cylinders are used for test and standard Nessler cylinder. Both are matched tube, clear, colorless glass with uniform internal diameter and having a transparent base. The transparent base or glass of the limit test apparatus nominally capacity of 50 ml. The overall height is 150 millimeter. The external height to the 50 ml mark at the Nessler cylinder. In the limit test apparatus, as we see, the apparatus is called Nessler cylinder with a nominal capacity of 50 ml. Total height is 150 millimeter along with the external height up to the 50 ml of mark. The limits for impurity, which are specified in a monograph of the pharmacopoeia, the limit of impurities fixed for individual pharmaceutical substance, but the quantity to be tested for limit test in that use directly sample, either it may a liquid or a solution as directed in the monograph. If test sample made to fail in the limit test for particular impurity and add small quantity of standard impurity into sample and perform the test, the turbid solution is non-clear solution and the opalations change in the color. Select identical natural cylinder keeping next to each other. Difference between 50 ml mark of both cylinder should not be more than 1 millimeter. As we see in the figure, the diagram of natural cylinder, we see the total if mark of 50 ml along with a 1.5 3.0 millimeter in diameter. First we discuss the limit test for chloride. Limit test for chloride to limit the amount of chloride as impurity in inorganic substance. As we see in the reaction, the chloride with the silver nitrate lead to form silver chloride. The soluble chloride present as impurity in the limit test for chloride. As we see the principle behind limit test for chloride based on reaction of sodium chloride with silver nitrate in the presence of dilute nitric acid to form silver chloride, which appear as solid particle in the solution. In this figure, as we see the reaction previously, in that the procedure like addition of 1 ml silver nitrate, then distilled water up to 50 ml, 1 ml of the nitric acid, 150 ml of the sample in standard. The addition of 10 ml known impurity is about 5 ppm. Procedure for the limit test, we have to take two natural cylinders. They are labeled as test and standard. So in the test we see the addition of sample in 25 ml of water and then add to the natural cylinder. On the other hand, we have used a standard solution of chloride with 25 parts per million in the natural cylinder. In both, we add a 10 ml of dilute nitric acid and make up the volume up to 50 ml. Both to add 0.1 m silver nitrate solution about 1 ml. Both are stirred immediately with the glass rod, allowed to stand for 5 minutes, then observe the reaction. Both are protected from the light, view transparently against a black background. In the observation we see opalations produced in sample solution should not be greater than standard solution. If opalation produced in sample solution is less than the standard solution, the sample will pass limit test of the chloride. In the reaction we add the nitric acid. Nitric acid added in the limit test of chloride make solution more acidic, help silver chloride precipitate to make solution turbid at the end process of the reaction as insoluble silver chloride. As we see in the limit test for sulfate, this test is designed to determine the allowable limit of sulfate contained in the sample. The principle behind the limit test for sulfate is the reaction of a soluble sulfate with the barium chloride in presence of dilute hydrochloric acid to form the barium sulfate, which appear as the solid particle show turbid of the solution. Potassium sulfate has been added to increase the sensitivity of the test. The observation of sulfate limit test is the barium sulfate reagent contains barium chloride, sulfate free alcohol and small amount of potassium sulfate. In the observation, the turbidity produced in the sample solution should not be greater than the standard solution. If turbidity in sample solution less than standard solution, the sample be pass the limit test of sulfate. There are various reasons in the limit test of sulfate. The addition of hydrochloric acid, it helps to make solution more acidic. Potassium sulfate used to increase the sensitivity of the test by giving ionic concentration in the reagent. Alcohol helps to prevent supersaturation, produce a more uniform opalescence. The procedure as we make a test and standard solution. In the test, add 10 ml of 25% solution of barium chloride. The addition of ethanolic sulfate standard solution, both are mixed properly, allowed to stand for 1 minute. The given sample dissolve in 15 ml of water and 0.5 ml of acetic acid to the solution of natural cylinder. Both stir immediately with the glass rod and allowed to stand for 5 minutes, then view transversely against a black background. The limit test for arsenic. Arsenic is an undesirable harmful impurity present in the medicinal substances. All pharmacopoeia prescribe a limit test for arsenic. The pharmacopoeial method, which is based on the Guget test. In this, special reagents are used for limit test of arsenic, marked and distinct by letter T, means they all should be arsenic free and should themselves confirm the test for arsenic. The principle behind the limit test of arsenic, in that this reaction is based on the reaction of arsenic gas with hydrogen ion to form a yellow stain on mercuric chloride paper in the presence of reducing agent like potassium iodide. This is also known as Guget test. The special precaution should be used. In the limit test of arsenic, the first point is the arsenic acid which is present in sample, that is reduced to arsenious acid by adding the reducing agent like potassium iodide, stannous chloride, zinc, hydrochloric acid. That means arsenious acid further reduced to arsenic gas by hydrogen, then it reacts with the mercury chloride paper which give a yellow stain. The depth of yellow stain on mercury chloride paper will depend upon the quantity of arsenic which is present in the sample. The reaction as shown in the figure. There are certain cautions which should be undertaken while performing the limit test for arsenic. When we dissolve the sample in acid, the arsenic present in sample gets converted to arsenic acid by the addition of reducing agent like potassium iodide or stannous chloride. The arsenic acid gets reduced to arsenious acid. The third point, the nascent hydrogen formed during the reaction. Further, it reduces arsenious acid to arsenic gas, which react with the mercury chloride paper and it will give a yellow stain. The arsenic limit test apparatus, as we see, the arsenic converted to arsenious acid. This arsenious acid with the nascent hydrogen produce arsenic gas. Then arsenic gas is compared. Then the mercury chloride will show the stain. The intensity of stain is mentioned in the pharmacopoeia which gives the limit for the arsenic. The apparatus we use in the arsenic, a wide mount glass bottle capacity 120 ml with 2.5 cm of the diameter at the mouth. Then bottle fitted with a rubber bung which passes a glass tube. The tube is constricted at its lower end and about 1 mm diameter, and the upper end of the glass tube fitted with two rubber bungs. Each have a hole bored centrally exactly 6.5 mm in diameter. After that, as we see in the figure about the apparatus for arsenic, both bungs are fitted to the upper end of the tube. The first bung in such a way that mercury chloride paper gets exactly sandwiched between the central perforation of the tube. The rubber band or spring clip which used as a manner that the gas evolved from the bottle must pass through a 0.65 mm internal circle mercury chloride paper. As the procedure we previously see, known test solution prepared by dissolves specific amount in water, then add stannated hydrochloric acid, then potassium iodide, graduated zinc, keep solution for 40 minutes to boil the gas. Now, few reasons are there against the arsenic limit test. These reasons are the stain obtained on mercury chloride paper compared with standard solution. The standard solution or the standard stain must be freshly prepared. It will be fade while on keeping. The inference of this test is if the stain produced by test is not deeper than standard stain, the sample may comply with the limit test for arsenic. The addition of stannous chloride used to complete evaluation of arsenic, zinc, potassium iodide and stannous chloride used as are reducing agent. The addition of hydrochloric acid make the solution acidic. The lead acetate paper used to trap any hydrogen sulfide. The stannated hydrochloric acid. If pure zinc and hydrochloric acid are used, the steady evolution of gas does not occur. This will produce improper stain. So, to get steady evolution of gas, stannated hydrochloric acid is used. The use of lead acetate solution. Hydrogen sulfide gas may form during the experiment. Zinc contain as sulfide as impurity. It gives a black stain on mercuric chloride paper. So the gas evolved are passed through cotton wool plug moistened with the lead acetate. The use of potassium iodide. Potassium iodide is converted to hydrogen iodide which bring reduction of unreacted pentavalent arsenic to trivalent arsenic. This reproducible result can be obtained if it is not used, then some pentavalent arsenic remain unreacted. The limit test for iron is based on the reaction in ammonical solution with the thioglycolic acid in the presence of citric acid to form iron thioglycolate. In other words, we also it called the ferrous thioglycolate complex which produce pale pink to deep reddish purple color in alkaline media. Thioglycolic acid is used as a reduction. In the reaction, the color of ferrous thioglycol complex fade in the presence of air due to oxidation. Color destroyed in presence of oxidizing agent and strong alkali. The purple color develops only in the alkaline media. So ammonia should be used. The ammonia react with iron, form precipitate of ferrous hydroxide. So citric acid is used which prevent precipitate of iron with ammonia by forming a complex with iron as iron citrate. The procedure as we see previously, the same test solution and standard solution prepared and add to the Nestle cylinder, addition of 20% weight by volume of iron free citric acid. After that add the thioglycolic acid. Dilute with 50 ml water, allow to stand for 5 minutes and observe color. The observation color produced in the sample solution should not greater than standard solution. If purple color produce sample solution less than standard, the sample will pass limit test for iron. The reason behind the addition of citric acid. This will form a complex with metal cation and help precipitation of iron by ammonia by forming a complex. Thioglycolic acid helps to oxidize the iron. Ammonia added to make the solution alkaline. The pale pink color visible only in the alkaline media. The limit test for heavy metal designed to determine content of metallic impurity are colored by hydrogen sulfide or sodium sulfide under the condition of test should not exceed heavy metal limit given under the monograph. The metallic impurity or heavy metal impurity, these are maybe iron, copper, lead, nickel, cobalt, bismuth, antimony. Limit test for heavy metal indicated in the individual monograph in term of parts per million of the lead. That means the parts of lead per million part of substance being examined. The heavy metal impurity like heavy metal expressed as quantity of lead required to produce a color of equal depth as in standard comparison solution with definite quantity of lead nitrate. Quantity stated as heavy metal limit expressed as parts of lead by weight per million part of the test. The remain distributed in colloidal state give rise to brownish color. IP method or Indian pharmacopoeia give a limit for heavy metal that is 20 part per million. Indian pharmacopoeia adopted three methods like the applicable for the sample which give clear colorless solution under specified condition of the test. In the method 2, the sample do not give clear colorless solution under specified condition. In the method 3, use for it substance which gave clear colorless solution in sodium hydroxide medium. Limit test for lead. Lead is the most undesirable impurity in medical component. Comes through use of sulfuric acid, lead line apparatus and glass bottle use storage of the chemical. The principle behind the limit test for test is it is a reaction of a lead and dithiazone in alkaline solution to form lead dithiazone complex which is red in color. The reaction which will be take place in the limit test of lead. Dithiazone in chloroform able to extract lead from aqueous alkaline solution as dithiazone complex which is red in color. The original color of dithiazone is a green color, but in the presence of lead thiazone it gives a violet color. The resulting color at the end of process is read intensity of color of the complex depend upon the amount of lead in the solution. The lead dithiazone complex in chloroform compared with the standard volume of lead solution treated the same manner. The impurity in this gets separated by extracting an alkaline solution with the dithiazone extraction solution. The methods which is used is transferred by a separating funnel. In that 6 ml ammonium citrate with potassium cyanide and 2 ml of hydroxylamine. Then add 2 drops of the phenol red. Solution made alkaline by adding the ammonia solution. Then combine thiazide extract shaken for 30 seconds. Then 30 ml of nitric acid and chloroform layer is discarded. Then add 5 ml of diathiazone standard solution similarly as previously. The observation of this test, the intensity of color of complexes depend upon the amount of lead in the solution. The ammonium citrate or potassium cyanide, hydroxylamine hydrochloride used to make pH optimum. So interference and influence of other impurity has been eliminated. The phenol red is used as indicator to develop color at the end process. The addition of ammonia in the lead limit test is added because in the limit test of lead sulfide produced by addition of standard hydrogen sulfide to the solution contain lead. The pH level is 3 to 4 necessary for precipitation of a lead sulfide. So aqueous ammonia or acetic acid is added to maintain the pH level. Thanks a lot